Welcome to this lesson on photosystems, the remarkable light harvesting complexes that power photosynthesis. Photosystems are specialized protein structures found in the thylakoid membranes of chloroplasts, and they play a crucial role in converting light energy into chemical energy that plants use to produce food. In this video, we'll explore the two types of photosystems and understand how they work together to capture sunlight and transform it into the energy molecules that fuel all plant life. Let's begin by defining what photosystems actually are. Photosystems are large protein pigment complexes that are embedded within the thylakoid membranes inside chloroplasts. These complexes are made up of proteins combined with light-absorbing pigments, primarily chlorophyll and carotenoids. Their main job is to capture light energy from the sun and convert it into chemical energy during the light-dependent reactions of photosynthesis. Think of them as tiny solar panels within plant cells that harvest sunlight and use it to drive chemical reactions. There are two distinct types of photosystems working in plants: Photosystem II and Photosystem I. Interestingly, they're named based on the order in which they were discovered, not the order in which they function. Photosystem II contains a special chlorophyll molecule called P680, which absorbs light most effectively at a wavelength of 680 nanometers. Photosystem I contains P700 chlorophyll, which absorbs light best at 700 nanometers. These different absorption wavelengths allow plants to capture a broader range of light energy from the sun. Photosystem II has a sophisticated structure with four main components. First, the antenna complex acts like a satellite dish, containing numerous light-harvesting proteins packed with chlorophyll and carotenoid pigments that capture incoming light. Second, the reaction center houses the P680 chlorophyll pair, where the actual charge separation occurs when light energy is absorbed. Third, the oxygen-evolving complex is responsible for splitting water molecules, which releases electrons needed to replace those lost from the reaction center and also produces oxygen as a byproduct. Finally, the electron transport chain transfers the energized electrons to plastiquinone molecules, beginning their journey through the photosynthetic process. Now let's walk through how photosystem II actually functions. Step 1. When light energy strikes the antenna complex, it gets funneled to the P680 chlorophyll molecules in the reaction center, exciting electrons to a higher energy state. Step 2. These highly energized electrons are immediately transferred to a primary electron acceptor molecule, preventing them from falling back to their ground state. Step 3. The P680 chlorophyll is now missing electrons, so the oxygen-evolving complex splits water molecules to provide replacement electrons. This water-splitting reaction also releases oxygen gas and hydrogen ions. Step 4. The energized electrons then move through an electron transport chain embedded in the thylakoid membrane, eventually reaching photosystem I while generating ATP along the way. Photosystem I has a similar overall architecture to photosystem II, but with some key differences. It also has an antenna complex that harvests light energy using chlorophyll and carotenoid pigments. The reaction center contains the P700 chlorophyll pair instead of P680, which is where charge separation occurs in this photosystem. Photosystem I features iron-sulfur clusters, which are metal-containing proteins that accept and transfer the high-energy electrons through the complex. Finally, it has a ferredoxin binding site on its outer surface, where electrons are transferred to the enzyme NADP plus reductase, which will ultimately produce NADPH. Here's how photosystem I operates in the light reactions. Step 1. Photosystem I receives electrons that have traveled from photosystem II through the electron transport chain. These electrons have lost some energy during their journey. Step 2. Light energy absorbed by the antenna complex re-energizes these electrons when they reach the P700 chlorophyll in the reaction center, boosting them to an even higher energy level than before. Step 3. These high-energy electrons are then transferred to ferredoxin, a small iron-containing protein that acts as a mobile electron carrier. Step 4. Ferredoxin delivers the electrons to the enzyme NADP plus reductase, which uses them to convert NADP plus into NADPH by adding electrons and a hydrogen ion. NADPH is a crucial energy-carrying molecule used in the Calvin cycle to produce sugars. The beauty of photosynthesis lies in how these two photosystems work together in perfect coordination. Photosystem II initiates the entire process by splitting water molecules and starting the flow of electrons through the system. Photosystem I then re-energizes those electrons to produce NADPH, which carries chemical energy in a usable form. Together, they accomplish three vital outcomes. Oxygen gas is released as a byproduct when water is split. ATP is generated as electrons flow through the electron transport chain between the two photosystems, and NADPH is produced to carry high-energy electrons. These products, oxygen, ATP, and NADPH, are essential for both plant survival and all aerobic life on Earth.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.